So as we just mentioned, membranes are selectively permeable. They will allow certain molecules to move across the membrane on their own, and others will not be able to pass on their own. So large polar molecules and ions are going to have a hard time passing through that hydrophobic interior of the membrane. Now, that doesn't mean that these molecules can never cross a membrane. They certainly can. They just need the assistance of what we call a transport protein. So transport proteins are embedded in the membrane, and they basically form an alleyway or a channel for these molecules to move across. Some of them are carried across, some of them are just kind of passively moving through from one side of the membrane to the other. But basically what the transport protein is, and you're looking at one here in this kind of light greenish bluish color here, is just an alleyway. It kind of walls off that hydrophobic area. So this part of the protein would be hydrophobic, but the interior would be hydrophilic, and it would allow those molecules to pass across. All right, so we have receptor proteins that receive chemical signals that are embedded in the membrane. We have transport proteins that allow molecules to move across the cell membrane. There's also what are called recognition proteins, which act like name tags on the outside of a cell to allow one cell to identify a cell from another cell, or at least to identify that it's part of your body and it's not some pathogen that has invaded. So there's a wide variety of different functions for these proteins that are embedded in the membranes. Now that we've talked about the construction of a membrane, let's talk about how materials move across a membrane. So there are different mechanisms for how materials move, and we're going to start with what's called passive transport. So passive transport refers to the fact that no energy is required to move these molecules across the membrane. We're using what we call a gradient, or a difference between two regions, as the driving force for the movement of these molecules. So the first type of passive transport is what we call diffusion. So diffusion can occur with any type of molecule, and we say it's the tendency of a substance to move down its concentration gradient. And it's moving from an area where the molecule is much more concentrated to an area where it is less concentrated. So a membrane doesn't necessarily have to be involved with diffusion. We can talk about opening a perfume bottle, right? So inside of perfume bottle, there's all these perfume molecules. And as we open up the perfume bottle, you can start to smell the perfume because these molecules are moving around, they're bouncing around inside of the bottle, and every once in a while one of them will bounce to the outside and you can smell it. So it's moving from where it's more concentrated inside of the bottle to where it's less concentrated outside of the bottle. So we call a concentration gradient, once again, we're looking at the concentration of the molecules, where they're more concentrated and where they're less concentrated. And that gradient just refers to that difference between those two areas, the difference between inside of the bottle, our perfume bottle, and the outside of our perfume bottle. In our image on the right, it's showing a membrane. And we've got a carbon dioxide molecule, and we can see that there are a lot of carbon dioxide molecules on one side of our membrane and very few on the other. If our membrane is permeable to our carbon dioxide molecule, then the carbon dioxide can diffuse across the membrane from where it's more highly concentrated to where it's less concentrated. Another type of passive transport is what we call facilitated diffusion. So we've already mentioned that transport proteins are required to move ions and large polar molecules across the membrane. So facilitated diffusion simply means that our transport protein is going to help to facilitate the movement of our molecules across a membrane so that they can move down their concentration gradient. So what do we mean by down a concentration gradient? We mean it's kind of moving with its gradient. It's still moving from where it's more highly concentrated to an area where it's less concentrated. So if we think of our membrane as separating two areas, one above and one below, and then we have our blue transport proteins here, and this one is also a transport protein in yellow. They're just doing it slightly differently. We've got potassium ions. The K with the positive is for potassium. So we've got these orange circles here that are highly concentrated on one side. They're going to move through the transport protein to the opposite side. So moving from high concentration to low, and we're simply doing it through our transport protein. So this is facilitated. Same thing with amino acids. These are slightly larger molecules that are polar. We're going to use our transport protein to move from a high concentration to a low concentration. And this is just showing calcium. So once again, we've got another example of an ion that's going to move from high concentration to low through a transport protein. This is still passive transport. No energy is needed for this to occur. Our last form of passive transport is what we call osmosis. So this refers to the movement of water. So with diffusion, we can talk about any molecule moving down its concentration gradient. With osmosis, we're only talking about the movement of water, and we're saying it's the diffusion of water. So that means water should move from where it's more highly concentrated to where it's less concentrated. And we're going to see water moving across our selectively permeable membrane. And once again, this is still passive. No energy is required for this to occur. Now with osmosis, we like to use some terms that describe our solutions on either side of our membrane. So you can kind of think of them as adjectives. We have the term hypertonic and hypotonic. So hyper refers to more. So we're referring to a solution that has more or a greater solute concentration as compared to another solution. So greater solute. So the solute is whatever is dissolved in our solution. So I have the example here of tap water compared to distilled water. Distilled water has been put through a process to remove the impurities in the water. Tap water has all kinds of things that have been added to it to make it smell better, make it taste better. So there's a lot of things that have been added to tap water, and those are removed in distilled water. So tap water would have more solutes compared to distilled water. So we would describe tap water as being hypertonic compared to distilled water. So if we're saying that one solution has greater solutes, we have to say it has greater solutes compared to what? So it's always we're comparing it to some other type of solution. Now we might have the opposite scenario where the solution has less solutes than another solution, and that is what we describe as being hypotonic. So a solution with lesser solutes compared to another. So the example in this case is tap water compared to seawater. Seawater has a ton of different things in it, right? First off, it's got a lot of salt in it, right? It is salty. So there's all kinds of different salt ions. There's also gold that is dissolved in seawater. There's actually a small amount of gold in seawater. Not enough that you would want to dry out a whole bunch of seawater and see if you could make a gold bar out of it, but still, it's in there. So tap water would be considered to be hypotonic compared to seawater. So tap water has less solutes compared to seawater, which has more. And lastly, we have the term isotonic. Iso refers to equal. So a solution of equal solute concentrations. So they're about equal to each other. Now I have this phrase here that water moves from a hypotonic solution to a hypertonic solution. So there's two ways to kind of remember how water moves during osmosis. We can say that water moves from where it is more highly concentrated to where it is less concentrated, or we can look at the solute concentration and say that water moves from a hypotonic solution to a hypertonic solution, one with greater solute concentration. So let's look at this. At the top here, let's say we've got this container and these dashed lines in the middle represent our membrane. These little reds are oxygen with the two, looks like Mickey Mouse ears almost, are our hydrogen. So these are all water molecules. The gray rings represent a solute that is dissolved in our water solution. Our membrane is only permeable to water and not these larger ring-like solutes. Notice the water level on the top is even on both sides of our membrane. So we want to predict how water will move by the process of meiosis. Will it move from the left side of the container to the right side, or will it move from the right side to the left side? Well, if you think about it, we said osmosis is the diffusion of water, and diffusion is the movement from an area where something is more highly concentrated to where it's less concentrated. So where is the water more concentrated? It is concentrated on the right-hand side, so it will move from where it's more concentrated to the left side where it's less concentrated. And that's what we see in the figure below, that the water level on the right-hand side has dropped and it has increased on the left-hand side because the water has crossed from the right side to the left side. Another way to think about this is to look at the solute concentration. So if we describe our solutions on either side of our membrane using our hypertonic and hypotonic, the right side of our membrane would be hypotonic because there's no solutes, compared to the left side where there are several solutes that are in there. So this would be described as being hypertonic. So the left side is hypertonic, the right side is hypotonic. And we know that water will move from a hypotonic to a hypertonic solution. I always think of this as sweet tea. So I lived in North Carolina for a long period of time, and sweet tea was extremely sweet. If you think about it, sweet tea has a lot of sugar in it, and sugar is a solute. So if we think of these as all sugar molecules in our sweet tea, in order to be able to drink the sweet tea, because it's way too sweet, it's too sugary, you might want to add water to it to dilute it so that you can drink it. All right, so you're adding water to your sweet tea, or you're adding water to your hypertonic solution. All right, and that allows us to predict the direction of the movement of water. So once again, you can think of it as water's moving from where it's more highly concentrated to where it's less concentrated, or water is moving from a hypotonic solution to a hypertonic solution. We are adding water to our sweet tea, which has too many solutes in it, too much sugar.